Hey everyone! In the last few videos, you looked at the different subdisciplines of genetics and what they study. Now in the next few videos, you'll learn about model organisms and the important role that they play in efficiently determining gene function. As usual, as you go through these videos, remember to fill out the learning guide, and by the end, you should be able to answer 2 questions.
And our first question is, how can we determine the function of human genes without experimenting on humans? And our second question is, how do the characteristics of model organisms help determine gene function? Studying genes in humans presents technical, ethical, and experimental problems. So, how can we determine the function of human genes? One approach is to find a species that is easier to study, like a mouse, and that also has most of the same genes as humans. Here's a graphical representation of part of the human and mouse genomes. We can see that the genes humans have are also present in the mouse. If we do an experiment in mice and determine that the ICAM2 gene plays an important role in the immune system, we can infer that the same gene in humans could possibly do the same thing. So by studying an easier to study model organism, in this case the mouse, we can start to determine the function of human genes. A model organism is an easier to study species used to learn about species that are harder to study. This is possible because they have many of the same genes. Fortuitously, around 75% of genes involved in human genetic diseases are also found in model organisms. Studying model organisms has led to important discoveries about human diseases, including cystic fibrosis, diabetes, and multiple sclerosis, or MS. And you'll see several examples of this throughout the course. The mouse is the model organism with the most genes in common with humans, but other model organisms are very often used to study gene function for other hard-to-study species, or to study specific processes, like cell division, in detail. We'll look at these model organisms next. 6 commonly used model organisms are the bacterium Escherichia coli, or E. coli, the fungus Saccharomyces cerevisiae, or baker's yeast, the tiny worm Cyanorhabditis elegans, or C. elegans, the fly Drosophila melanogaster, which is Morgan's fruitfly, which I'll just call Drosophila, the plant Arabidopsis thaliana, or just Arabidopsis, and the animal Mus musculus, or the mouse. My lab and I study Arabidopsis, so forgive me if I tend to talk about that model a bit more. You'll learn about the particular strengths of these model organisms and what they're used to study in the reading, so be sure to check that out. We'll approach model organisms and their usefulness in the following way. First, we'll look at the characteristics of humans, which will appear at the upper left, and we'll look at why these characteristics make it harder to study human genes, which will appear at the lower left. Characteristics of model organisms will appear at the upper right, and why these characteristics make them useful for studying genes will appear at the lower right. Now, annoyingly, humans like to choose who they mate with. So if Johnny here likes Jane, we can't just say, hey Johnny, go over and mate with Rachel, and in several years, let us know what all of your offspring look like so we can see how your traits are inherited. Ethically, forcing humans to mate to produce offspring and study them is generally frowned upon. Though I don't know why Johnny wouldn't mate with Rachel. She has this very nice red hat. Anyway, that means that we can't do controlled genetic crosses in humans. Fortunately, most model organisms don't mind if we force them to mate with certain individuals. Well, okay, if they do mind, we're sort of willing to do it anyway in the name of science. So we can do controlled crosses in model organisms and mate a chosen individual with another chosen individual and analyze the offspring to determine, for example, which phenotype is dominant, much like Mendel did. Mendel crossed a homozygous plant with purple flowers, and let's say the genotype of this plant is P1P1, to a plant with white flowers, and let's say that its genotype is P2P2. Now, since the offspring get one allele from each parent, all the offspring of this genetic cross are heterozygous, or P1P2. If all the offspring are purple, then P1 must be dominant, and of course the purple phenotype must be dominant. Nowadays, we use Arabidopsis instead of pea plants because it's smaller, easier to grow, reproduces quickly, and because we can manipulate its genes so we can study them in detail. Not only can we use genetic crosses to determine what phenotypes and alleles are dominant, but also to do things like determine the distance between genes, study gene interactions, and, as Mendel and Morgan did, to study the principles of genetic inheritance. Another reason humans are hard to study is because we generally only have a few offspring, and it takes 9 months for each child. Who wants to wait around for 9 months to get the results of a cross? Plus, humans take many years to reach the age where they can reproduce. In other words, they have a long generation time. So, if we want to study traits present in the next generation, we have to wait a long time. Many applications of transmission genetics that we've gone over before require that many offspring are produced from a single mating or from the same parents. Even if we could force people to mate and reproduce their entire reproductive lives, and again, not okay to do this, they would likely only produce 15 to 20 offspring, and this isn't even enough for most of these applications. Fortunately, model organisms can produce many offspring at once and reproduce quickly. In other words, they have a short generation time. Arabidopsis, for example, can produce around 10,000 tiny seeds from a single plant in about 6 weeks, which is its generation time. So if you see one of these in your yard, pull it out right away or you'll be fighting weeds for years. Drosophila takes about 10 days to reproduce and produces hundreds of eggs. Getting lots of offspring from crosses was important for Mendel in particular because in order to come up with his 2 laws, he needed to observe the ratios of different phenotype combinations in the offspring of his crosses. For example, this 9:3 to 3:1 ratio. If he only got like 5 offspring from a cross, this rare offspring type might not have appeared, and this would have confounded his experiment. Mendel grew and observed thousands of offspring in order to rigorously test his hypotheses. So having lots of offspring is important for studying how genes are inherited. Even today, in eukaryotes like Arabidopsis and Drosophila, Gene mapping requires hundreds or even thousands of offspring. In gene mapping, a heterozygote is crossed to a homozygous recessive individual. How often the different genotypes appear in the offspring can tell us the distance between these two genes. If we only had a few offspring, this wouldn't be possible. So model organisms having lots of offspring allows us to determine the distance between genes.